As a continuation of our talk on the application of different biomolecules in nanobiotechnology, today we will discuss about bacteriorhodopsin and its potential technical applications. So, bacteriorhodopsin is a retinol protein molecule which is found in the photosynthetic system of a salt marsh bacterium called Halobacterium salinarum. Retinal protein. So, this is a protein which is found in the human eyes, right? And this is a chromophore. Chromophore is a molecule which absorbs light at a certain wavelength, and the color of a molecule is responsible. It is responsible for the color of the molecule, or what we observe a color of a compound is due to the presence of a retinal. So in its native form, the bacteriorhodopsin molecule is located in a cell membrane commonly called as a purple membrane. So the surface of the halobacterium salinarium actually contains patches, membrane patches, which are called as a purple membrane, the PM. This purple membrane is made up of proteins and lipids. And we find that the ratio of proteins is about 75% and lipid is that of 25. So the predominant protein which they found in this purple membrane was your bacteriorhodopsin, and it is found as a two-dimensional crystal. Okay, so this is formed by the bacteriorhodopsin trimers. So the bacteriorhodopsin is found, to sum up here, the bacteriorhodopsin is found in the purple membrane, and these purple membrane are the membrane patches which are found on the Halobacterium salinarium. So these purple membrane contains proteins and lipids, and which is about the 75% of the protein is about bacteriorhodopsin, and this bacteriorhodopsin is a retinal protein. Now, what is the function of this bacteriorhodopsin in the cell, in the bacterial cell? Within the bacterial cell, what is its role? So, within the bacterial cell, it is critical to the survival of the organism in an oxygen deficient environment. Okay, so the major function of a bacteriorhodopsin molecule is to act as a light driven proton pump. Its major function is to the major function of bacteriorhodopsin within the cell, it acts as a light-driven proton pump. Fine? It acts as a light-driven proton pump, which transfers the protons across the membrane, and this generates a proton gradient, right? Which in turn produces an electrochemical potential, which is used to synthesize ATP. So, effectively, the bacterium uses bacteriorhodopsin to convert the absorbed sunlight into chemical energy. The absorption, so actually what happens, the absorption of the sunlight initiates a photocycle. Okay, we'll discuss about this shortly. It initiates a photocycle which is accompanied by the transportation of protons. Now, what is this photocycle? Now, the characteristic features of this photocycle only we are going to utilize for the development of optical sensitive films, right, and further other technical applications in your nanobiotechnology. Now, when you come back to the structure of bacteriorhodopsin, as you know, we are talking about the bacteriorhodopsin which is extracted from Halobacterium salinarium. Fine. This is made up of 248 amino acid residues and in a polypeptide chain which consists of 7 transmembrane helixes. The structure is that of a 7 transmembrane helixes. So each Br molecule contains a chromophore named retinol. We know retinol is the major protein present in your bacteriorhodopsin, which is covalently linked to the amino group of lysine 216 in the G helix via protonated Schiff base. Okay, so this Schiff base is located in the center of a cavity enclosed by the 7 membrane helixes. We said that it is a 7 membrane helix. In between the or at the center of the 7 membrane helix, you have a Schiff base. Okay, this is what which effectively divides the proton channel into the two partitions, the extracellular side and the cytoplasmic side. Okay, so the extracellular region and cytoplasmic region mostly contains charged residues or hydrophobic residues, which serves as a proton donor to the Schiff base. Now the next slide will see the detailed structure of the Bacteriorhodopsin. So this is the schematic representation which shows the structure of the Br molecule within the purple membrane. Fine. So here you can see that the structure of the 7 membrane helix of the Br molecule, and at the center of the cavity you have the protonated Schiff base, and to this Schiff base you find the attached retinol. Fine, retinol. Fine. So the red arrows, what these red arrows and the direction, they are, these red arrows indicate the movement of the proton. Fine. And the numbers also indicate, if you look into the diagram, the numbers 5, 3, all this indicates the number of the protons. Okay, so here you find that from the cytoplasmic side, from the cytoplasmic end to the extracellular region, what happens, the proton is translocated, and for this translocation you need the assistance of this, Br molecule. This Br molecule, as you have seen, is a 7 membrane helical structure which contains a protonated Schiff base in between, which is connected to the retinal molecule via the lysine 216. Fine. So the two kinds of retinal chromophores exist in the Bacteriorhodopsin. Okay. So the Bacteriorhodopsin, the retinal chromophore, as we have talked about, they absorb the wavelength, certain wavelength light. So they exist in all trans and 13 cis conformations. So what we saw, the structure right now, is the structure which exists in the dark condition, the normal conditions. In the dark condition, the retinal chromophore moiety of Bacteriorhodopsin is all trans, which is bound to protein via the Schiff base, right? This is what we discussed just now. But under light stimulating conditions, all trans retinal is converted to the 13 cis retinol. Okay, so this isomerization, we call it as the isomerization from the trans to the cis conformation. So which is induced by the light. That is why it is called as photoisomerization. So the photoisomerization of the retinol chromophores, okay, what does it do? It indicates a photochemical reaction. Okay, so it induces a photochemical reaction in the Bacteriorhodopsin proteins, and it is a complex process which involves 7 intermediate stages. Okay, this is what we call it as a photocycle. So you have 7 intermediate stages with distinct spectral absorption maximum called the photocycle of Bacteriorhodopsin. So the Bacteriorhodopsin, how long, actually it usually just spends 15 microseconds on translocating, right, on translocating a proton from the cytoplasmic side to the extracellular side of the membrane. Okay, so upon your light stimulating condition, that is upon your presence of sunlight, induction by sunlight, when the Bacteriorhodopsin is induced or absorbs your light, okay, it becomes all trans becomes cis conformation. During this transformation, what happens, this undergoes 7 intermediate stages. Okay, and actually, so while translocating protons, what happens, they undergo 7 intermediate stages, fine, which have distinct spectral absorption maximum, and the total time taken for this process is just 15 microseconds. And during this photocycle, not all the photo intermediates can go back to the ground state. Okay, so once after they have translocated the proton, not all of them go back to the ground state. What happens, certain compounds, or sorry, certain states remain like irreversible stages. There are certain irreversible stages in the photocycle, and also there is a branch within the photocycle. Now these two are the major, like what you say, these are the two major factors which are of consideration, which are of interest in our photocycle: the branched photocycle, and also the production of certain intermediates which are irreversible stages. Okay, these two are the characteristic features of the photocycle. Now in the next slide, we'll look into the actual schematic representation of the photocycle. This is a schematic representation of the photocycle. So under the stimulation of light, fine, under the stimulation of light, your Br molecule. So here this subscripts, the numbers indicate the absorption maximum, the wavelength at which they absorb maximum. Okay, so 570. Okay, then you have 570. Then you have it undergoes a change into the, it changes into the J molecule. Okay, K, L, M, N. Okay. O, right? So what happens? These are the intermediate stages, and this is a branched cycle. This is a branched cycle. These are the intermediate stages. Now we find that actually how long each exists. So 8 microseconds, 2 microseconds, right? So the, and this is just 15 milliseconds, right? So this is just your 15 milliseconds. So what happens is you have the stage, irreversible stage. Okay, so MEC to the MCP, this is a irreversible stage. Now, how you are going to apply this bacteriorhodopsin for your nanobiotechnological applications? Okay, so this is now processed, or this is accomplished, this is attained by coating this bacteriorhodopsin, or preparation of this bacteriorhodopsin in as layers. Okay, so you are preparing bacteriorhodopsin layers on various substrates. Okay, so oriented bacteriorhodopsin layer is needed in the integrated hybrid devices to generate sufficient photoelectric signal. Now we have seen that what are the unique features of the photocycle. Okay, the photocycle is induced by the light, and you have the production of ATP. Okay, so there is a photocurrent which is generated. Now this is the feature which you are incorporating in your hybrid devices, that is in the normal electronic devices, right? Electronic devices, you are incorporating a biomolecule. Here the biomolecule is your bacteriorhodopsin. So this is a, what you mean as a hybrid device. Okay, the goal of the bacteriorhodopsin orientation technology is to orient the protein effectively and prevent the purple membrane from denaturation. Okay, we have to see to that the hybrid device which you are going to design contains the bacteriorhodopsin in a proper orientation, and it also should be safe. It also should be protected from your denaturation. Okay, so there are different technologies which have been introduced to reconstitute the bacteriorhodopsin layer on various substrates. Okay, so here we will be talking about 4 different methods. One is the Langmuir-Blodgett deposition, electrophoretic sedimentation, biotin streptavidin mediated monolayer, antibody mediated monolayer, electrostatic absorption. Okay, so we'll see the schematic illustration over here. Okay, so the first one, the first diagram or the first illustration on the left shows the electrophoretic sedimentation of PM. PM means the purple membrane onto a membrane, onto a film, PVA film. Okay, so here how is it done? You have, it's an electrophoretic setup. You are applying 4 volts, okay, to the PVA film for 5 to 10 minutes, and the PM patch was deposited at the bottom electrode with supernatant appeared on the, with supernatant appeared in the upper layer. So here you have the purple membrane suspension. Okay, so after application of your 4 volt, what happens? Now it gets deposited in this membrane. Fine. The second method is your Langmuir-Blodgett deposition. So here the bacteriorhodopsin is deposited onto the solid substrate by the applied electric field. Fine. And the third one is your simplified structure of a stable purple membrane deposited by streptavidin biotin mediated monolayer technique. We know this receptor ligand, okay, biotin streptavidin affinity. So through this you are stabilizing the PM on a monolayer. Fine. And the last one also shows the stabilization of BR layers on various other negative charged surfaces. Okay, so these are the schematic representation of how you are going to deposit the BR layer on various substrates, okay, which can be used in your hybrid devices, in your electronic devices. Okay, why we term it as hybrid? It is being a incorporation of the bacteriorhodopsin on existing electronic devices. Fine. So here we come to the photochemical application of bacteriorhodopsin. Okay, so once you have known the structure, till now we have known the structure of the bacteriorhodopsin and how you are going, what is a photocycle, how it is going through the 7 different intermediate stages and how a photocurrent is produced. Okay, and now we saw how these bacteriorhodopsin membranes or the layer is deposited on various devices. Okay, so here the first application is a photochemical application. Here what they did, the researchers proposed the use of bacteriorhodopsin as an active layer in a single write-in optical memory. Okay, so the optical memory, where the optical memories, they incorporated this, the bacteriorhodopsin layer. So the writing of the data was done by the pulsed doubled lasers. Okay, so laser is used to write the data. Now the data is written on a substrate which contains bacteriorhodopsin. Okay, so the reading of the data stored in the BR film can be achieved by a conventional optical microscope, right? An optical microscope is enough, or directly by a charged coupled device, CCD, right, or a CCD line camera. Okay, charged coupled device. So through this, the laser is used to write the optical memory, and you are using an CCD matrix to read the data which is present in the BR active layer. So what is this? The incorporation of biomolecule. The second application, the incorporation of the biomolecule into hybrid semiconductor based holographic associative processes. Okay, you can incorporate the BR layers on the holographic associative process. So this can be useful in your real time computing. Fine. Now the next one, photoelectric application. Okay, so actually the principle which you are using is the light driven proton pump. Okay, bacteriorhodopsin light driven proton pump. Fine. What does it do? It detects changes in the light. It detects changes in the light. It detects changes in the it detects changes in the light just like a human eyes. Okay, so but the differential photocurrent response, okay, that is what gives you the information, okay, of the moving objects. This can be incorporated in the motion biosensors. Okay, what is motion biosensor? It is a visual information system, okay, based on which light sensitive biomaterials such as your bacteriorhodopsin, okay, is incorporated, and it can detect the or sense the optical flow to detect the information of motion of objects. As the name suggests, motion biosensors are the sensors which detect the movement of an object. Okay, biosensor, because we are incorporating the BR layer in this. Why this is of interest? BR layer can detect the changes in the light. Okay, so they can detect changes, but it is reflected. Okay, it is reflected as a response, photocurrent response, which is useful for detecting the movement of the objects. Fine. This is the BR based photocell, the structure of a BR based photocell. Fine. So here you have, it is a sandwich layer. Okay, it is a sandwich layer. As you can see, this 1 and the 5. Okay, so this is a sandwich structure. 1 and 5 are the glass substrates. Okay, so 1 and 5 glass substrate, 2 and 4 are the iridium tin oxide transparent coating. Okay, these are the coatings, and your 3 is the polyester thin film, and in between you have the deposited BR film. Okay, so this is the biosensor system which detects the movement of an object. Okay, this is just a schematic representation. This photocell is used to detect the movement of the object, right? Photovoltaic cells for the, right? So when the light stimulates on the surface of Bacteriorhodopsin, we have seen that it induces a series of photochemical reactions resulting in the Bacteriorhodopsin pumping protons, right, from the intracellular side to the extracellular, and it also leads to the production of the photoelectric response. Okay, so researchers are utilizing this application and mechanization of the produced photocurrent, okay, for production of the photocurrent. So studies on the Bacteriorhodopsin hybrid composite materials or sensors, right, are based on the inorganic and polymer materials and different operations. Okay, so from enhanced proton transfer to the electron transfer effect. So we incorporated the Bacteriorhodopsin layers onto the photovoltaic cells for enhanced proton transfer to electron transfer. So we are mechanizing the production of, for the mechanization of these devices, we are incorporating the photovoltaic cells. Fine. And one more application, Bacteriorhodopsin related immunosensor. Okay, so this was first based to detect, okay, this was the Bacteriorhodopsin based photoelectric immunosensor was demonstrated for direct label free sensitive microbial detection. Okay, you can detect microbes with the help of this immunosensor. Now what the working methodology is that the antibodies or affinity immobilized on the purple membrane coated electrode. Okay, so the antibodies are coated on the affinity immobilized purple membrane electrode, and the photocurrent generated by the antibody coated sensor was reduced after incubation with E. coli cultures. Okay, so you are incorporating incorporating the electrode. Okay, they are incorporating the BR layers on the electrode, and this electrode is used for sensing the, or used along with the culture for sensing, right? So as there is an increase in the growth, okay, as there is an increase in the growth of the bacteria, what happens, there is a reduction in the level of the photocurrent which is generated. Okay, so this is due to the bacterial light shielding effect. Okay, so the bacterial light shielding effect will reduce generation of the photocurrent, and this reduction in the photocurrent is measured, and this gives you the like indication or quantification of the level of the bacterial growth. Okay, so the photoelectric immunosensor could achieve the quantitative single step detection of up to 10 to the power of 7, okay, CFU per ml microbial cultures. Now, the Bacteriorhodopsin, and very important biomedical application is the Bacteriorhodopsin artificial retinal prosthesis. Bacteriorhodopsin has been explored as an important photoactive element in the protein-based retinal prosthesis. Okay, so for the patients who are suffering from retinal degenerative diseases, the researcher who pioneered this work is Michael Friedrich. Okay, he produced a simple color-sensitive artificial retina. Okay, in this artificial retina, BR, that is your Bacteriorhodopsin, and your two artificial variants were used to form a matrix as photosensitive elements. Okay, so this was very easier to come for construction, and its performance was also high. Okay, so what he did was he employed an artificial neural network in the system to learn self-organizing color adaptation algorithm. That is, upon your light stimulation, what happens, there are certain changes on the Bacteriorhodopsin. Okay, so this color change, right, it's converted into a algorithm to tune color space transformation, right, and and this employs an artificial neural network. Okay, so in this system, the optoelectrical signal which is generated by the elements consisting of the 3 proteins. Now, what are these 3 proteins? One is bacteriorhodopsin and the other 2 other photosensitive artificial variants. Okay, so these 3 proteins will generate an optoelectrical signal which is amplified, and this amplification is recorded by a computer, right, that contains or that is stimulated as a pre-processing layer. Okay, and owing to this bacteriorhodopsin was photochemical or photoelectric properties, all right, in this system, the system was regarded as an intelligent color sensitive artificial retina. So successfully they have utilized this use of Bacteriorhodopsin incorporated retinal processes. Now here it is the schematic representation of this artificial retinal processes. Fine. Here the artificial neural network, a neural network algorithm detects the output. Once light falls on the sensor, okay, once light falls on the sensor which contains your bacteriorhodopsin and 2 other artificial variants of this protein, there is a signal which is generated. This signal is now recorded by a computer, right, and it is processed. So this helps the unsupervised learning, right? Fine. So this is of a great use in your artificial retinal processes. So fine. So till now we have discussed about the structure of the bacteriorhodopsin protein. Okay. So it is a 7 helix transmembrane protein, right, which contains a Schiff base which exactly divides the channel into the cytoplasmic side and then your extracellular side. So upon light induction there is a photocycle which is initiated and production of ATP, and this ATP production is associated with the 7 stages, intermediate stages of the bacteriorhodopsin, and there are few stages which are irreversible stages, and it also involves a branched cycle. Okay, so now these properties of your bacteriorhodopsin enhances or enables its use on a hybrid devices. Okay, so biohybrid electronic devices. Okay, so this field we term it as a bioelectronics, which merges biology with the electronic technique. So here we have seen few very major applications like the synthesis of bacteriorhodopsin layers for detection of your microbial cultures as an immune sensor, for detection of your motion, right, of an object, movement of an object, for detection of your electrophotovoltaic cells, and also in artificial retina, right. With such wide range of applications, bacteriorhodopsin still can be utilized, or still it is being explored for the application of your biohybrid electronic devices. Fine. Hope you understood this. If you have any doubts, please leave your feedback and we'll get back to you soon. Thank you.